while she's setting up the next talk, what I want to do is very quickly, last two people that I want to introduce you to is Lauren, our medical student extraordinaire, who does a lot of our work, a lot of our quick statistics, quick review of data, et cetera, in the natural history study team. And then last but not least is Nikki. And I don't know, Lindsay may be the only one that knows Nikki in here. I don't know. But anyway, Nikki is actually the lead coordinator on every single one of these studies that I'm going to be talking to you about, okay? So she knows all the data. I didn't include a disclosure slide here, but let me just suffice to say that, again, I mentioned that we have, Richard mentioned that we've done up to 10 trials in the complement inhibitor space. We try to do them all. That's how we try to reduce our bias, is see if we can do everything instead of just one and choose a favorite sort of thing. But suffice to say that I do have conflict. I do have disclosures because I do have the clinical trials that we do run here. So it's completely, in that way, completely separate from moral and what their basic science research is. So we still have the same question. Are we there yet? As we're journeying on our way to success. And so for this one, we're going to review the anti-complement drug landscape. We're going to review the current availability of clinical trials. And we're also going to discuss next steps. And again, what I want to share very quickly, I mentioned my disclosures, but I also want to share that I cannot technically share anything that's confidential data. I can't share something that has been shared with Richard or I by any one of these pharmaceutical companies. I can only share with you what's public data. And I want that to be very clear because I don't want to get into trouble with anybody. Both of these big phase 3s have been shared in the nephrology world. So they've been shared with other nephrologists either in Stockholm just recently or in Argentina just recently, but they've not been shared to the public, the patient public, if you will. So they will be new to the patient public, but I want everybody to know that they are not confidential data, okay? Because I don't want to get into trouble. So let's start with a very quick reminder, and this is just meant to be a reminder because you guys already know this, what the drivers of disease are. Roughly 21% are in genes that are, again, in the middle of the pathway, but then a larger number of patients will have those nephritic factors. The only reason to show this to you is because, again, we're talking about that is where the activity of this disease appears to be for the vast majority of patients, and that is why that area of the alternative pathway became the target for a number of pharmaceutical companies. So here was the first one, and Richard, you might have to remind me, but you presented this back in 2011 at one of the complement conferences, and so this was the very first complement inhibitor that we actually trialed here at the University of Iowa, and this is the start of my little bit of history for you. So 2011, you can see it was first presented. Then Yuzo, who I think you probably have met, then he actually presented not only in vitro, but in vivo data. This was a single patient that we actually tested for this medication. And this is a soluble CR1. And to orient you, soluble CR1 should inhibit C3 or C3B. So that's the area of the pathway that we're talking about when we first tested this initial, this drug. And based on what I just told you, where the activity of this disease appears to be, that sounds like a good choice, right? Or at least I hope it makes sense. Then they actually opened a clinical trial in 2013. So we began enrolling patients in 2013, but then this happened. Unfortunately, they shut down the CDX-11-1135. It wasn't a safety issue. It was, you guys may appreciate this. It was too hard to recruit patients. There weren't enough patients. It was too rare of a disease, et cetera, et cetera. They found that it wasn't, and I got this off their website. I didn't get this from their.
This was sure one other thing. It was really hard to make it. So they didn't have very much. And so they wanted us to recruit light patients.
Yeah, so we couldn't find enough rare skinny patients, I guess, was the problem. So Anyway, so you can see that, you know, this was back in 2011 when we started down this journey, right? So here's your next one. We had lots of hope about this one, Amy 101. Some of you that have been coming to these meetings for a while have known about this one because we've talked about it forever. Also, it's a, actually, I'll show that to you. It's a C3 inhibitor, and we had lots and lots of hope about this medication. And in fact, they even announced that they were going to be doing an Amy 101 trial in C3G. Then absolutely nothing happened for us. So again, we have yet another sponsor who scientifically, it sounded like this would be a good choice for us, but in fact, they never launched an actual C3G study. They are active in the space. They are a complement inhibitor. They are doing other things, but it just didn't work out to be correct for them to be doing it in the C3G space. So then the next one for us actually became avacopan. That was the next study that Nick and team were involved in. And this one is a C5a receptor blocker. So with this one, you're blocking, by virtue of being the receptor blocker, you're blocking the function of C5a. So you're blocking a aflatoxin activity. And in fact, this is the data which has never been published. We're hoping that we will see it at some point. And I don't know, you guys won't be used to looking at clinical trials, but what you're hoping for in p-values is 0.05. So if you compare placebo with treated group here, that's almost a 1 p-value. That means for their primary endpoint, they had practically no difference between the treatment group and the placebo. And this was actually in C3G patients. There were some good aspects of this trial. We'll have to wait for the publication to actually find out what those were. But suffice to say, this drug moved on to another disease that was actually very C5a. C5a was a good target for that disease, and actually they've done well. I don't know if they'll come back into our environment for the C3G or not. Not published, already mentioned that. Okay, the next one was this drug called danicopan, which actually was a factor D as in dog blocker. And if you, again, remember where the pathway is, or if you recall, and I don't remember whether we mentioned it specifically, but D is necessary to cleave B so that you can get a BB that becomes part of the convertase. So again, this drug actually makes some sense, but unfortunately, though the title looks nice, we're presenting the final results. The bottom line is it didn't work. So there was no difference between the placebo and the treated group of patients. And in fact, if you dig in deeper, there were probably some pharmacokinetic issues. It may not be that the drug's not going to work. It actually maybe could work in a different circumstance. But again, this happened in 22. So I've tried to do this chronologically for you. This was published in 22. We were doing the study 20 through 22. And in fact, another agent that we were trying to get off the ground wasn't able to get off the ground. Then we go, I only bring this one up because I often tell you all to look at clinicaltrials.gov, and you will see this one on clinicaltrials.gov. So that's the main reason I'm bringing it up. We actually are not doing the study here in part because it's not actively recruiting, but also because for this particular one, I had to bring up the bigger pathway that Richard presented this morning. We're no longer talking about that amplification loop area of the alternative pathway, if you will. We're talking about all the way over here in the lectin pathway. So it's a different target. We don't know yet whether it will work for C3G, but it's the phase 2 of this is ongoing, and it was what they call a basket study. So they're doing some IgA, some lupus patients, and some C3G patients all in the same study. They're trying to collect information, obviously, to try to figure out whether it makes sense. It says on clinicaltrials.gov that they're intending to do a phase 3 for C3G in 2025. So we'll keep you posted. I don't know whether that's going to happen or not, but we'll keep you posted. And just for you to know what it says again on clinicaltrials.gov, it will be the adult patients, so greater than 18-year-olds. And then the standard, all of these studies have required greater than 1 gram of urine protein and a definite C3 glomerulopathy diagnosis to actually get into the trial. Okay, So let's just very quickly, before I introduce the two phase 3 trials, I'm going to go back to this pathway. The major site of dysregulation is the AP. I can't say that enough. It's either at the C3 convertase or at the C5 convertase. It remains unclear still how much of the terminal pathway, so C5b through 9 or MAC, if you're used to that terminology, is actually involved. So again, that center area of the pathway is where these phase 3s were meant to target. So that's again why they were predicted to be a reasonable approach to this. So it just wasn't willy-nilly that we thought that they would actually be a good choice. So there's your pathway, but let me present to you the animal models that actually support the possibility that these drugs could be good. And the first one is, this is the one that everyone that or most people who work in the C3G world now use in their laboratory setting, because it's the mouse model of C3G. It happens to be a C3GN, so it's not DDD, but a C3 glomerulonephritis mouse. That's the one that most everyone will use in their laboratory to study this disease. I need that as your background so that I can tell you that if you also knock out C5 in that mouse that has C3GN, they get improvement, but they don't get complete remission of their disease. Why is that significant? Because that mouse model would be your equivalent of being treated with terminal complement blockade, right? That mouse model should represent what being treated with terminal complement blockade does. And in fact, that's certainly worked out to be the case in humans for a number of humans. What if you knock out complement factor B in a mouse model that gets C3GN? It turns out that that mouse model's disease goes away completely. So you can see that this wasn't just based on, we guessed it should happen, that, you know, targeting the alternative pathway or that middle area would be good. There's actually animal evidence to support that. And then finally, this one's a little more convoluted, and hopefully I don't confuse you, but there wasn't I couldn't find a good mouse model for this one. So this one is actually a rat. Hopefully you can see the difference. But this one is, and this is actually to represent this idea, if you block C3, or let's say you just deplete it completely or make it not useful for the body, it turns out that you can clear the C3C. That's what we happen to see on the biopsy. You can clear the C3C off the tissue, off of the kidney biopsy, and actually it happens in 24 hours. So that's really impressive, that if you were to either block C3 or completely deplete it with something, that you actually might have some improvement, because we assume that that deposition is a problem, right? So clearing it off the biopsy should be a good thing. So in fact, these two animal models were in part responsible for us understanding that blocking B and blocking C3, C3b should be a good idea. So now we have, sounds right for the human example in terms of where the nephritic factors are and where the animal models are. So now let's go ahead and start into them. Factor B inhibitor, B is in boy, it's, you can see where it is there. It's right in that center part of the pathway. This happens to be an oral, and there are people in the room that actually have been on this one and still are on this one. It's oral, it's twice a day. It's phase 2 included both native and transplant. That's only important because their phase 3 did not include the transplant part. They already had their data from the phase 2, so they didn't happen to include it in their phase 3. But their phase 3 has included the adults, and the adult phase 3 is completely done. That's the one that I'll give you the data on. The adolescent one is the one that I told you is enroll 5 globally, watch for a while, enroll 5 more, et cetera. So the adolescent part of that one is still going on. It included a 6-month placebo. So this is what regulatory agencies require in order for drugs like this to actually make it to approval. And I already mentioned that the adolescent arm is still recruiting. So I'm just going to give you some really fast slides on this. And so I didn't share with you what their primary endpoints were, what their secondary endpoints were, but suffice to say, in the nephrology world, we always worry about urine protein. We worry about whether the GFR or the creatinine is stable. So those are going to be featured heavily in both of these that I'm going to present to you. But these studies also have looked at things that I think that are potentially even more important too, things like fatigue and things like that. So I'll very quickly introduce these things. And again, this is only the publicly available data. This isn't the data that we presume will be seen by the regulatory agencies. Suffice to say, the first point is that iptacopan, also called Fabhalta, that's the drug that blocks factor B. And in fact, the 24-hour urine protein was significantly decreased. That's the blue area here. So by 30.2% in the iptacopan treated group of C3G patients, versus there was actually an increase in the urine protein for the placebo group. And again, this was statistically significant. This is also important for a nephrologist, and this is kind of a complicated slide, but really nephrotic range proteinuria, so the really high urine proteins greater than 3 grams, are the ones where almost everyone will swell, be swollen, have edema if you have greater than 3 grams. But importantly for us, the more urine protein you have, the sharper your slope is to end-stage kidney disease. So it is believed to be an important prognostic indicator if we can get your urine protein down below nephrotic range. That's the background. I've skipped a few important pieces there, but really what I want to show you is that if you think about the group, so the dark box here, if you think about the group that in iptacopan, they started at 55.3 and they went to 31.6 patients who actually had nephrotic range proteinuria. So definitely down, as opposed to if you were in the placebo group, you went from 30.6 up to 41.7. So really all I want you to see is you don't have to record the differences obviously in your head, but just to say that in the treated group, they actually had an improvement in the number of patients that had nephrotic syndrome. And in the placebo group, it actually got worse, the number of patients that actually had nephrotic range proteinuria. And then, this might have been a little bit more exciting. Well, let me just say very quickly that it's even better if your urine protein and your GFR move in the right direction. That's really what the point of this is supposed to mean. And really, you were 7 times more likely to have both of those things happen to you if you were in the treated group versus if you were in the placebo group. That's really all I want to share with you on this one. The GFR also increased in the treated group versus it decreased in the placebo group. The caveat here, because I impressed this issue upon you when we were talking about avacopan, this was not statistically significant. But still, this is a good thing, that you actually had an improvement in the GFR in one group and not in the other. This particular slide always makes me sort of happy. This is what was happening to the GFR before the patient joined the clinical trial. And that's what I was telling you. It was going down. This was the 2 years before they entered the study. And then this is what their GFR looks like once they enter the study. So this is all comers. And so again, from a nephrologist standpoint, this is a win. This is what we would like to see. We would like to see that if they're on it even longer, the GFR keeps going up, right? That would be even more, more important to see. But only 6 months was the amount of that placebo control. And then this, less impressive, but I think even some of you have actually even reported this to me that there was an improvement in the degree of, I should reverse that so there was less deposition of C3 on the biopsy, not quite like our rat model that we were talking about earlier, but it was a little less, if you will, in the placebo group. So again, not statistically significant, but we're beginning to see a signal that actually the C3 deposition is improved in that group that's exposed to iptacopan. And again, recognize that this was a very short trial. So we may see it improved even more if they were on longer. And then unfortunately, also not statistically significant, but there was a decrease in the fatigue in the iptacopan treated group, as opposed to, and this doesn't surprise you all, in the placebo group, so the group on regular therapy or standard of care, they actually had a worsening of their fatigue. And then the biomarkers are actually as expected. They were actually, they either went up or down based on what we would expect if you shut off the complement system in that setting. And then importantly, because, you know, I'm going to run out of time here, but importantly, the iptacopan was safe. So the final statement is that there was no deaths, thank goodness, but there are also no cases of meningitis or meningococcal sepsis for this group of patients. And like eculizumab, the risks for these new drugs is encapsulated organisms. So everyone does need to be vaccinated for all those encapsulated organisms. And that, again, that's something we can talk about offline. But generally, what you should perceive from this moderately busy slide is that iptacopan was in fact safe. So now let me move on a little bit faster, or see if I can get this done in 5 minutes. This is the C3B inhibitor. They just completed their phase 3, so they did their press release on August 8th. In fact, the reason I can present this data because it was presented publicly to the nephrologist at the called GlomCon, happened to be in Maui this year. They presented this data in GlomCon just recently, so that's why we can present it to you. Only thing I really want to point out here is that two things. First of all, it's a subcutaneous infusion given twice a week. So this is a different delivery mode. This is, and that may make a difference to some of you, but also importantly, this study, and I don't know if I highlighted it with my patients 12 years and older. So this one automatically reached down to the 12-year-old. So they have their data already, where the other group is still waiting just a little bit longer. Included C3G and immune complex GN. That might be important for some of you. And it included both transplant and native kidney disease. So this study did basically all of them. So that's why I think I'll be able to sort of speed through this for you a little bit. But the bottom line is that they saw a 67.3% improvement in the urine protein in the treated group. No improvement in the placebo group. Importantly, and that's what these next couple of slides are going to be, I mentioned all those different groups of patients in the study. It didn't make any difference if you had C3G or you had immune complex GN. You still got a very nice improvement in your urine protein. That's really the main point of this slide is that we now know that you can treat both of those groups of patients and still expect to get some improvement. It also didn't make any difference if you were an adolescent, so 12 to 18-year-old or an adult. You still got a very nice improvement. It probably doesn't surprise you that adolescents did just a little bit better. That probably is because they've had their disease a little less long, perhaps. So maybe the proteinuria wasn't quite as fixed. That would be a guess on my part, but that might be the case. Or finally, it didn't make any difference if you were native kidney or transplant kidney. You responded to the medication equally well. Transplant kidneys, I shouldn't say equally well. Transplant kidneys, this is an excellent number, by the way, but as Dr. Thomas would, I think, mention to you this morning, transplant kidneys do, they have challenges. But still, we would definitely consider this to be an important improvement. So what's next in this journey? Well, first of all, for that particular study, we need to see all the other parameters that you saw in the iptacopan study. I need to see what happened to the fatigue. I need to see what happened to the biomarkers. I need to see all the other pieces. Those have not been released publicly. They will be released at the American Society of Nephrology meeting coming up at the end of the month. So after that, I'll know the answer to the rest of those questions, or at least I'll be able to publicly be able to speak about it. So with this data in mind only, what's next? Well, we need regulatory review of these. We need peer review. So my colleagues out there in the nephrology world need to review this and figure out whether this data is as good as it looks, compared to some of this, to what I've shown you. We need to complete the studies in the down to 12-year-old, but we even need to now go further, right? We need to be able to get down easily to the 7 or 8 year old probably before we really can say we've done the best we can do in children. So that's still a considerable distance ahead of us, if you will. We need to achieve those regulatory approvals, not only have them reviewed, but actually get a regulatory approval. Then we need to design the protocol. The current KDIGO that says that everybody gets mycophenolate mofetil and steroids, is that going to be the same? Or are we going to say one of these agents is the current first agent? We just have to find out whether that is. We need to define those use protocols, whether it's in children or in transplant, et cetera. We need to go through all of that so that regardless of who the nephrologist is who's treating you, it's that straight line treatment approach. We can hopefully be able to treat you with the best, with some of the best data we have. And then of course we have to monitor to make sure that we're actually doing what we think we're doing with this group of patients. And oh, by the way, does this treatment effect last the lifetime of the patient? That would be really wonderful to see. Thank you.